Scholars Conference providing this platform to express and to showcase our research work conducted. So to introduce, Dilip has already introduced, I'm Manohar, presently working as assistant professor from the Department of Pharmaceutics, NGSM, Institute of Pharmaceutical Sciences, India. This work was conducted at JSS Academy of Higher Education and Research in the JSS College of Pharmacy, Mysore in India. And since finishing this work, I've moved on to NITTE or NGSM Institute from past 2 months. And I thank both these institutions, one for providing a platform to present this work and the other to conduct a research. So today, in brief, to tell about, I'm going to present the research work conducted during the PhD research work, which was on fabrication and evaluation of 2 bioactives, that is quercetin and beta-carotene, into a nanoemulsion form for improving its oral bioavailability and therapeutic efficacy in diabetes. So, keeping the time constraints, I have summarized the contents or the presentation into these 5 headings. That is, we see a brief introduction, followed by the materials and methods, then see results, summary, conclusions, followed by few references. So to moving on with this introduction, let me take to a brief about the diabetes. We know that diabetes is increasing day by day as a chronic metabolic disorder, and it is predominantly characterized by hyperglycemia, that is increased level of blood glucose level. So recently, even the International Diabetes Federations has revealed that there are already 382 million adults or patients, youngsters, way back in 2013, who are affected with this diabetes, and which the number might go on to reach 592 million by the end of 2025, that is another next 2 years, which promoted us to get interested and to contribute towards improving the patient compliance, or to contribute towards the management of diabetes management in our own knowledgeable way. So here, with that aspect of selecting diabetes, then we went on to see, are there not any other medications available? Yes, there are. But why is it not a diabetes treatable? We all agree that diabetes, there is no permanent cure, but can be managed well with various activities, including the exercise, diet, and medications. So let us concentrate on the medication part of it. So the discovery and development of potential therapeutic agents to prevent the delay or the inception of diabetes is being of great clinical significance in the recent times. So with that, and many synthetic drugs which we looked on for a research was having side effects. So one or the other side effects, so which made us to look into the way of bioactive compounds, which have been extensively utilized to prevent diabetes for its potential therapeutic benefit in the recent times. And also they provide a least toxicity, unlike the synthetic medications. So with that, one such molecule which we chose was a quercetin, which belongs to the flavonoid category, and it is known to inhibit a xanthine oxidase by reducing free radical generations, and thereby it prevents lipid peroxidation, which is one of the major cause for the oxidative stress, which in turn is the reason for the recently developing diabetes as reported. So why only quercetin? Why not any other flavonoids was the question. So, but on a review or the literatures, we got to know that among all the other flavonoids, quercetin has a potential therapeutic benefit in the diabetes mellitus due to its superior hypoglycemic activity, and its various mechanism of action is being under research in the recent times. So the report also suggests that quercetin not only has a hypoglycemic activity, but also it has insulin sensitizing activity through its various mechanism of actions. But this quercetin, as a pharmaceutical scientist, I looked in different perspective. That is, this quercetin with a huge therapeutic benefit against the diabetes mellitus possesses some limitations, and there are some bottlenecks to get the maximum potential benefit out of this quercetin. What was it? That was its formulation or druggability. It is not soluble, it is poor, it is hydrophobic in nature, and it has poor gastrointestinal absorption, which results in poor oral bioavailability. So this was the reason why we picked up a quercetin as one of the molecule. And coming to the second molecule, that is beta-carotenoid, which is a recent trend, we can say that as a formulation scientist, for its most abundance in nature. The beta carotene is abundant in most of the carotenoids present in food, and its therapeutic benefit in the carotenoids, especially in the diabetes, has already been proved for its both protective and therapeutic activity. So the potential health benefits of both quercetin and beta carotene were understood, and we also understood that these molecules lack bioavailability, lack oral bioavailability, and uptake and absorption was less. Hence, these two active ingredients were of keen interest. But now next comes to the picture was through what is that delivery system we can deliver these two molecules in a better way. So with that efforts, we looked on as a novel drug delivery systems. One such was a lipid-based delivery system, and specifically a nanoemulsion-based drug delivery systems, because these nanoemulsion-based drug delivery systems acts both as a pharmaceutical as well as a nutraceutical. This had that flexibility so that our formulation can be administered to the patients or can be suggested for the patients both as a nutraceutical as well as for the pharmaceutical purposes. And also these lipid based delivery systems, that is nanoemulsion systems, are known to improve the oral bioavailability and absorption of many active bioactive compounds, which was done for various other purposes. So hence we decided that we'll take up a nanoemulsions for delivering this selected drugs, that is quercetin and beta carotene. So the next step what was in front of us was how to prepare a nanoemulsions. What are the various methods? As we all know that there are two methods, that is the low energy methods and the high energy methods. And we chose high energy methods, which included the ultra-sonication, high pressure homogenization, and microfluidization technique, out of which we chose ultra-sonication for its benefits, because choosing this high energy method and ultra-sonication method enables us to use a lesser surfactant and then produces narrow ranged particle sizes, nano-sized particle sizes. So hence we chose with that. And with this, we came up to the objectives of our research is to primarily develop an select ideal nanoemulsion components. As we know that nanoemulsion components, we speak oil phase, the surfactant that together called as S-mix, and then the drug, its solubility, oil emulsifying ability, various things comes. So ideal selection of a nanoemulsion components for both quercetin and beta carotene was our first objective. And then second, to see and to evaluate, to characterize the developed optimized nanoemulsions for its improved bioavailability and antidiabetic property in the in vivo animal model. So with these two objectives, we moved on. And to brief the various methods, what we chose was, the first was the solubility analysis, where both this quercetin and beta carotene were checked for its solubility, maximum solubility in various nanoemulsion components, including the oil phase, the surfactants, and the co-surfactants. So how it was done? It was the active ingredients. I mean to say, I repeat that active ingredients I concerned for the quercetin and beta carotene together here. So the active ingredients taken up in a surplus amount was added to a minute amount of the nanoemulsion components. They were mixed to vertexed well, and they were placed in an isothermal shaker for a period of 3 days, that is for 72 hours at 25 degrees centigrade to attain equilibrium. So once after attaining the equilibrium, the samples were centrifuged so as to obtain only the supernatant, and then it was analyzed by using UV spectrophotometer at the 371 nanometer quercetin and beta carotene for at 461 nanometer respectively. This was the solubility. And the oil emulsifying ability. The solubility is one key area, we know that, but the second and the most important thing was the oil emulsification. The surfactant might have a very good solubility, and even in some cases, they did not to have a good solubility of a drug. But however, the surfactant selected for the nanoemulsion has to have a good oil emulsifying ability. Hence, as a reason, we selected few of the selected, means the surfactants which had a good solubility for both quercetin and beta carotene. We selected it further for oil emulsifying ability. This oil emulsifying ability was tested by taking 15% of the surfactant solution with water, and if 2.5 ml of this surfactant solution, we kept adding 5 microliter of the oil, that is the selected oil, and we vertexed. And this process continued until and unless a turbid solution is obtained. And finally, the amount of oil that can be consumed by this various surfactant solutions were observed, reported, and expressed as percentage, and hence called as oil emulsifying ability of the surfactants. And the next, the tertiary important component of a nanoemulsions are its co-surfactants. And these co-surfactants are selected to reduce the concentration of surfactant. That is one. Secondly, to improve the stability of the nanoemulsion. How do they do it? That is by reducing the interfacial tension between the two surfaces, water and the oil phases. They help the surfactants, we can say it in general. So the various co-surfactants were selected, and they were selected in a ratio of 1:1 with that of selected surfactants. And these 1:1 surfactants and co-surfactants were again combined with various ratios with respect to oil and checked for the nanoemulsion formation by making use of ternary phase diagrams. So these ternary phase diagrams helps us to observe that what the nanoemulsion region, or what percentage of, or how big nanoemulsion region can be obtained by using these composition or these ratios of surfactant and co-surfactants. And after this 1:1 ratio, we has to determine it. Is it only that 1:1 ratio, or whether there is any other further ratios can be modified? So here in the effect of S mix mass ratio, what we do is we keep the surfactant concentration constant. That is, for example, you can see it in the first chart, that is 1:0, 1:1, 1:2, 1:3, 1:4, 1:5. That means we keep the surfactant concentration constant, and by increasing the co-surfactant concentration. Thereby, once again, we go with vice versa. That is, we increase the surfactant concentration and keeping the co-surfactant concentration as a constant. And the same ternary phase diagrams would be developed to see what is that mass ratio of the surfactant and the co-surfactant mixture that would give us a good nanoemulsion region. So the phase diagrams which exhibits, so in a centrifuge tubes, when we keep adding with the water titration method, when we develop our ternary phase diagrams, whichever shows a transparent viable region, that we considered as a nanoemulsion region. So that was the one. And then we moved on to the next question, was preparation of nanoemulsion. And we know that we had selected ultra-sonication technique. So here what we did, we dissolved a drug in a predetermined amount of a surfactant, co-surfactant, and an oil phase. And then we subjected it for an homogenization at 5000 RPM for 5 minutes for both the drugs. Then typically it was transferred for ultrasonication using an vibrissyl ultrasonicator with the predetermined amplitude and time. So that we went on to optimize it further. And for the beta-carotene preparation, beta-carotene as an antioxidant was highly prone for oxidation. So we failed for its stability, or percentage drug content was on a question. And later on discovering, we went on to add a BHT, that is butylated hydroxytoluene, which is a synthetic antioxidant, which does not has any antidiabetic property, or it does not interfere, but only prevents the oxidation of beta-carotene. And we used it at 0.1%. That was the one. And moving ahead, after the preparation, the droplet size and the polydispersibility index of the nano droplet sizes were determined by using an instrument zeta sizes, that is 90. Whereas the particle size were identified by making a dilution with water at 1:100 ratio. The entrapment efficiency was also determined for the nanoemulsions by using centrifugation method. So by centrifuging at a higher RPM of 12,000 RPM for 20 minutes, we separate the nanoemulsion. Means we separate the emulsion systems, and then we filter it, and we take out the supernatant and we pass it through 0.2 micron or micrometer membrane filter, and the obtained solutions was identified or quantified by using HPLC with a developed method, and the percentage was expressed as amount of a drug content to the total amount of a drug into 100. The zeta potential, viscosity, pH are also an important parameters, and zeta potential was again identified, determined, or measured by using the zeta sizer instrument with a set of other separate cuvettes. whereas viscosity and pH were measured using the respective calibrated instruments at 25 degrees centigrade. And after the characterization, it was time to check for the stability studies. So after the preparations, we expect it to be stable with a wide range of temperature or the conditions. That is with 5 degree, 25 degree, 40 degree, and what we call it as accelerated condition, that is 55 degrees centigrade for 45 days of period of time. And within which we determined this droplet size and PDI for 0, 15th, 30th, and 45th day. So with the aim of the objective was to check for an improved release or improved solubility, which the drug release was the first step to check for the improved bioavailability. This acts as an in vitro evaluation parameter. So we compared here with that of a pure drug and our nanoemulsion, which is that which can have a highest solubility or which is that which can have a highest release. So to determine that, we had prepared a phosphate buffer, 7.2 pH, and 1% of surfactant was used just to ensure that the solubility of the drug in the medium. And this was used by using a dialysis membrane, a dialysis bag, wherein which the nano emulsions of a specific quantity was poured and sealed on both the ends. And later this dialysis bag was placed in a beaker containing a phosphate buffer solution, which is of 7.2 pH. And this dialysis membrane was of 12 kilodaltons, which was obtained from High Media, Mumbai, India. And the tubes were suspended, as I said, with a 75 ml of release media. And this release media was containing in a beaker, which was rotated on a magnetic stirrer placed at 37 degrees centigrade with 100 RPM. So there was a regular sample withdrawal, with sample withdrawal at regular intervals was done and was replenished with the same quantity of the fresh release media to retain the sink condition. The obtained samples were then checked or quantified by using the developed HPLC method. So because of the time constraints, I do not wish to about the method of HPLC what we used, but however, I would be pleased and obliged if anyone asks for it after the presentation. So it was all the characterization and the evaluation done. We were happy with the in vitro release, but now the real challenge comes in wherein we have to meet our second objective, that is improved bioavailability as well as the therapeutic efficacy in diabetes mellitus, for which the first step was anyhow, this objective was met with animal studies itself. The first was pharmacokinetic studies, wherein which we make use of male albino rats, Vista rats, which was around 250 to 300 grams were used. And this study was approved by the Institutional Animal Ethical Committee of JSS College of Pharmacy, JSS Academy of Higher Education and Research, Karnataka, India. So these animals were procured and they were into acclimatization for a period of 2 weeks. And after acclimatization, they were divided into 4 groups with randomly with 6 animals in each group. And all the rats were fasted overnight with access to only water before this experiments was done. So the dose was administered accordingly. So it was a single dose administration, which was given through an oral route. And in case of quercetin, it was 50 mg kg per kg body weight, whereas in terms of beta-carotene, it was 30 mg per kg body weight. Both the nanoemulsion as well as the pure drug were administered at a same dose. So after the administration, it was time for a withdrawal of blood samples, and blood sample of 300 microliters were collected from the tail vein into a centrifuge which contained 3% of sodium citrate solution. So the time interval was 0 hours, that is just before dosing, and with respect to 1, 2, 4, 8, 16, 24, and 48 hours respectively after the oral administration. So the collected blood samples were then centrifuged immediately. The plasma was separated and was stored at -80 degrees centigrade until further analysis by HPLC. So we made use of the same HPLC method developed to even determine the quantity from the plasma samples. And that was how we moved on to the in vivo anti-diabetic studies, wherein which even the same study was again approved by the same institutional ethical committee board. Here also the male albino visitor rats were procured, which were of 250 to 300 grams, were acclimatized, and with a humidity of 50 to plus or minus 5 degrees, 5% of humidity, with a 12-hour light and dark cycles condition for 7 days. So after the acclimatization, the house rats had food and water supply regularly. So after the acclimatization period, we induced diabetes by using streptozotocin, which is of 40 mg per kg body weight, and the STZ, that means to say that streptozotocin was prepared by using citrate buffer of 4.5 pH. So once it was injected intraperitoneally at 40 mg per kg body weight, we waited for 72 hours, that is for the 3 days of streptozotocin injection, and to check the blood glucose level. And once the blood glucose level was checked, and the blood glucose level, animals having more than 250 mg per deciliter of blood glucose level were considered to have developed a type 1 diabetes, and then those animals with a higher blood glucose level were randomly grouped into 4 groups, 5 groups with 6 numbers each as accordingly. So the normal groups, we did not had any STZ injections. They received 1.5 ml of regular water daily, and the control groups, they had a STZ induced diabetes, but without a treatment. But the standard group had a diabetes induced, and along with the metformin of a standard dose of 250 mg per kg body weight. So next comes our test groups, that is quercetin and beta-carotene, with a predetermined dose of 6.25 of quercetin and 7.5 mg of beta-carotene per kg body weight was selected and was given together. And the fifth group, that is the group E, was our keen interest, because as I said in the introduction, these ingredients might not be having a therapeutic effect, but what if they have a protective effect? That was a one question triggering in our mind, prompted us to go for this group to see whether these drugs had any protective effect or a therapeutic effect. So these groups were pre-treated for 21 days, or it was for 7 days with, and then it was induced with the diabetes, and then the treatment was continued. So just to see that how a pre-treated animals could respond to the STZ degradation of beta cells and could inhibit the damage or inhibit the onset of diabetes was one things to see. So what and all we measured was the body weight of the animals were measured regularly, the blood glucose level, and we measured oral glucose tolerance test. This is one wherein which the glucose tolerance keeps reducing once the diabetes has been induced. So we had checked it for a 10th and the 20th day. That is the median. The study was for 21 days. So we checked it for the 10th day and for the 20th day. Further, the biochemical analysis like total cholesterol, high density lipoprotein, glycerides, low density lipoproteins, the ratio of HDL, LDL, and VLDL was checked with the tissue injury markers. Because why this tissue injury markers was selected? Because the oxidative stress induced, STZ is known to induce diabetes by inducing oxidative stress. So this stress can also damage the various organs such as the liver and the kidney. Hence, we wanted to measure the tissue injury level, what was the extent that our drugs can protect the tissue injury. And then further we moved on to the histopathological analysis, wherein which after the 21st day of the experiment, the animal were euthanized and the organized were collected, stored in formalin, and observed for histopathological changes of both pancreas and the liver. So this was a short and brief the methods, and let us see now the results one by one. Moving on to the solubility analysis, wherein which I said that I would like to move in the same flow of the materials and methods were discussed. Here we can see that the solubility of both quercetin on the left hand side, beta-carotene on the right hand side were analyzed by using various nanoemulsion components. You can see a group of few numbers of oils, surfactants, and co-surfactants being taken. So here we had a propylene glycol, isopropyl alcohol, labrazole, transcotol P as the highest solubilizing agents in the terms of surfactants and co-surfactants, whereas it was ethyl oleate as an oil phase which had highest solubility of quercetin. Coming to the beta-carotene, it was a capsule as the highest solubilizing oil, followed by which the gelucide, aconun, and the labrafil as the surfactants and co-surfactants showed highest solubilizing power of beta-carotene. Coming to the oil emulsifying ability, we know that previous slide we have seen that it was a ethyl oleate which was we selected for the quercetin nanoemulsion preparation, and we subjected those selected surfactants for its emulsifying ability against ethyl oleate. So it was a tween 20 followed by the labrazole tween 80 span 85, and we could observe here the emulsifying ability was directly proportional to the HLB value of the surfactants. Tween 20 with the HLB value of 16 had a highest power of emulsifying ability, followed by the labrazole which had a HLB of 14, tween 80 followed by which had a HLB of 12, followed by span and labrafil. The same phenomena or the same similarity was observed even with the capmol, that is for the beta-carotene emulsion. Tween 20 with the highest emulsifying ability had a very good emulsifying ability, followed by gelucide and aconun. And then with this surfactants and the oil selection, we moved on to select a co-surfactant. So here we know that in the quercetin, we had seen that isopropyl alcohol, propylene glycol, transquitol P, and labrazole had very good solubilizing power of quercetin. But however, we had to know that whether these ingredients as a co-surfactants can help the tween 20, the selected surfactant, to reduce the interfacial tension as well as to produce a larger nanoemulsion region with that of. So this combination was of ethyl oleate and tween 20 was selected, and figure A, B, C, D represented a selection of isopropyl alcohol, propylene glycol, transquitol P, and labrazole. From the figure, we can see that it was propylene glycol in the figure B, which produced a significant, or not a significant, a good amount of nanoemulsion region with that of ethyl oleate and tween 20. But it was the D, that is the labrazole, which in combination with tween 20 at 1:1 ratio with ethyl oleate could produce a larger nanoemulsion region. Similarly, with the beta-carotene nanoemulsion components, we went on to check it with the capmol and the tween 20. Tween 20 was selected because it had the highest emulsifying ability. Capmol was selected because it has the highest solubilizing ability. So when we compared with these two 1:1 ratio with propylene glycol, transquitol P, and aconun, we could observe only a minimum amount of nanoemulsion region. So to be on the safer side and to improve our stability of our nanoemulsion, we went on to check the other surfactant. That is, we omitted tween 20 and we brought back gelucide, which had a second highest emulsifying ability next to tween 20. So this we could observe it as we moved on with propylene glycol, transquitol P towards aconun at 1:1 ratio. It gave us a nanoemulsion region. Hence, for the beta-carotene, we selected a capmol and a gelucide along with aconun as a S-mix mixture. Next, coming back to the effect of S-mix mass ratio, we just discussed in the materials and methods that it is not that 1:1 ratio of surfactants and co-surfactants we select, but we have to see whether there are any other better alternatives next to each other's lying for us to provide a good stable nanoemulsion. So here what we do is we just change the concentration of surfactant with respect to co-surfactant, and then change the concentration of co-surfactant with respect to surfactants. By doing such exercise, we could observe that at 1:4 and 4:1 of T20 and labrazole, we could achieve a good nanoemulsion region. That is figure F and figure I, which we could see a larger nanoemulsion region was observed with 1:2 ratio or 4:1 ratio. Similarly, in terms of beta-carotene, it is not similarly, it is contradictory. I can say that. As we can see in the picture A, B, C, D, E, as we move on alphabetically, the nanoemulsion region started decreasing unlike the previous exercise. So we had that 1:1 ratio of gelucide and aconun to be the best possible composition, to be the best possible mass ratio to give the beta-carotene nanoemulsion. So with this, we had went on to prepare the nanoemulsion with ultra-sonication technique, and we had optimized. And in the presentation, I had forcefully omitted with a disheartful omission of the optimization by box Benken design has been done to keep up the time. So after the optimization, the optimized formula was obtained and the optimized formula exhibited these results. We had a zeta potential of Quercetin nanoemulsion. We had a particle size of 127 nanometer with a PDI of 0.17, nearly ideally 2, which is very good, and with a zeta potential of -17 with a negative charge indicating that repulsive forces of the oil or the surfactants micelles formed, which was indicating that we might have a good stability. The viscosity was appreciable with 1.879 centipores and with a pH of 5.02. When we come to the beta-carotene nanoemulsion, we had slightly higher particle size of the optimized one with 153 nanometer and with a zeta potential of -14. The viscosity and the pH was little similar to it with 1.52 centipores and 5.82 pH. That was the one we had characterized. Now it was a time for the evaluation of the stability, how good our product would be stable. So for this, as I told, we stored our formulation with various temperatures of 5 degree, 30, 45, and 55 degrees centigrade. And we had evaluation regularly at 0, 15th, 30th, 45th day. So we could see that the bar graph on the left side is pertaining for the Quercetin nanoemulsion, and the graphs on the right hand side is for the beta-carotene nanoemulsion. The column graph indicates that the particle size, which is on the y-axis on the left side, the primary axis, indicated that the particle size tend to increase gradually, and we could see that at 55 degrees centigrade, there was a drastic change in the particle size from 125 nanometer to 225 nanometer by the end of 45 days. But however, it was observed that the 30 degrees centigrade was known to least get affected with that of particle size. Similarly, the dotted lines would indicate the PDI of the same, which indicated the similar response. That is the red color. You can see that there was a drastic change at 55 degrees centigrade with respect to PDI on the first 15 days. But however, it was maintained with respect to the 30th and 45th day. And at the end of 45th day, the PDI of the samples was almost similar. So we could conclude that there was no drastic change in the homogeneity of the particles or the droplets formed in the Quercetin nanoemulsion, but however, there was a maintained 30 degree, the sample stored at 30 degree maintained the PDI below 0.2, which was initially on the same. So yes, after this, yes, after this, I apologize. The heading says it's in vivo. It is an in vitro drug release studies where we had taken a dialysis bag, dialysis membrane, and we had checked at what rate the drug or the components as the nanoemulsion almost has reached 6,000 microgram per ml. So here, once we have reached 6,000 microgram per ml, which was given in 50 mg per kg body weight, it shows that it has more than 3 times the better absorption. Similarly, when in case of the beta carotene, it had almost 2,500 with respect to the pure drug, but whereas the nanoemulsion had reached a maximum of nearly 6,000, indicating that it has almost double time, twofold increase in the absorption. This led to us to take out the concentration for treating the diabetes mellitus for the in vivo anti-diabetic studies. So what conclusion we had drawn from this in vivo pharmacokinetics was, yes, of course, the nanoemulsion has a better absorption, and quercetin has a 4 times higher absorption, whereas the beta-carotene has 2 times higher the absorption. So hence what we had done was, the 4 times we had reduced the drug significantly. So 4 times indicating that it was 12.5 mg, it was as equal. And coming to the beta-carotene, where we have administered 30 mg, we had 15 mg. It was ideal. So in the animal dose, we had taken it almost a combination of 6.25 of what is that, quercetin, as well as 7.5 mg per kg body weight of beta-carotene. That is a 4 times lesser coming to with respect to the individual pure drugs. And then since we were going in combination, we further reduced the dose of the quercetin and beta-carotene to half of the 4 times reduced doses. That means we had taken only 6.25 plus 7.25 of quercetin and beta-carotene. As I told, we had groups classified into normal, control, standard, and then it came up to the treatment and a pre-treatment. The last group indicated with P in a brackets indicate that of pre-treated, wherein which the same drug at the same concentration were given 7 days prior to the experiment. Means it was pre-treated with these drugs for 7 days prior to the induction of diabetes. So the first measurement, what we keep doing on from day 1 to the day 21 was the body weight. And we know that the diabetes, it almost causes the decrease in the body weight. It drastically decreases, and we could clearly observe that the normal group had a steady body weight, steady or gradual increase in the body weight throughout the 21 days. But however, from the day 1 to the day 7, we can see that there was a decrease in the body weight in terms of control group, further which followed with 14 days and 21 days. But however, the reduction in the body weight was reduced or was inhibited by the standard, that is the metformin treated groups, as well as these treatment tested groups, that is quercetin treated groups, and this is a pre-treated groups. And we could see that by the end of 21st day, the pre-treatment group had significantly better inhibition of the reduction in the body weight with respect to the control group and the standard group. Coming to the about the by pre-treating the drugs before induction of diabetes. That is, even with the pre-treatment, the blood glucose level tend to be as normal as what of below 200 degree to below 200 mg per deciliter. It was quite significant in our study. That is what we could find it out in terms of blood glucose level between the pre-treatment and the treatment group. And coming to the oral glucose tolerance test, which was conducted on the 10th day and the 20th day. Figure A indicates that of a 10th day and figure B indicates that of 20th day. We could see a red line, that is a control group, and a black line passing down near to the axis, that is a normal group. But between these two were our tested groups laid on. And in both the cases, we could see that it was the pre-treatment group which had significantly improved the oral glucose tolerance test of the animals. Coming to the biochemical analysis, we had various analyses performed with respect to the total cholesterol, total glycerides, high density, HDL, LDL, and cholesterol to HDL ratio, HDL, LDL ratio, and VLDL ratio. So starting with the figure A, we know that the total cholesterol level increases with that of diabetes induction, but whereas with the treatment groups, it significantly was controlled. and it was potentially significantly controlled, inhibited in the rise of total cholesterol with the pre-treatment groups compared to that of normal treatment groups. Both were good, but however, both were significantly better with respect to the control groups. But however, there was no difference with the numbers. We can say that we cannot say that the significantly better result was obtained between the pre-treatment and the treatment group. But however, looking into the numbers, the pre-treatment groups had a lesser value compared to that of the treatment groups. Because the same scenario was obtained with the triglycerides level, wherein in a control group there was an increased triglyceride level, and all the other tested groups, including the standard, brought down this triglyceride, increased or elevated triglycerides level in the animals. So here also you could observe it very closely that there is no much difference between the pre-treatment and the treatment groups. And next, coming to the C, it was a HDL, and HDL tends to, high density lipoprotein tends to decrease with respect to the diabetes induction, but however it was restored and inhibited by the treatment groups. And however, it was slightly observed that here pre-treatment was better compared to that of treatment. It was not significantly better, we cannot say it significantly, but in the number or numerical wise, it was observed that we had a better performance of these pre-treatment groups. Coming to the LDL level in the diabetes increases, which is contradictory to that of HDL, and it was completely restored with respect to that of treatment groups with as comparison to that of a standard group. So we can say that, and lastly but not the least, it is a VLDL, that is a very low density lipoproteins. Even in that case, it was having a potential therapeutic benefit. So the outcome, or to summarize this biochemical test, what we could observe was, so what we could observe was the treatment and the pre-treatment groups were significantly better than to reduce these potential side effects or potential rise in the elevated in the biochemical parameters. But however, with the numbers to go on, we personally as a research scientist, we were little interested in this pre-treatment groups. So further moving on, I think I would like to conclude, tissue injury markers were also reduced with respect to the treatment and pre-treatment groups, and even the lipid peroxidation and superoxide levels were in control with respect to the treatment groups. Last but not the least, the histopathological regions, when we isolated the tissues and we could observe, we could see that there was a reorganized beta islet cells. There were prominent dark circled nuclei of hepatocytes, indicating that there were no damage, for significant damage with respect to the pancreas and livers for the groups treated with quercetin and beta-carotene in combination. However, the standard groups maintained the blood glucose level and all other parameters. The dilation of the hepatocytes, dilation or elongation of the beta islet cells was observed. And coming to the control, we could see the apoptosis in the liver hepatocytes, and the beta islet cells were ruptured, and the number of beta islet cells were found to be decreased with respect to the normal cells. So in conclusion, I would like to say that the quercetin nanoemulsion and beta-carotene nanoemulsion had improved therapeutic efficacy, and that is for sure. And one thing what I would like to mention over here is these two drugs were individually also tested for the anti-diabetic activity before we go for the combination. And in combination, these quercetin and beta-carotene has a potential valuable strategy for the management of diabetes mellitus. That is what we would like to propose, or we would like to suggest through this research. So this was a few references, and thank you. If there are any doubts, I would be happy to take on. It's over to you, Dilip.
Yes, thank you. Thank you, Dr. Manohar. Any questions from audience?